Hello everyone, welcome to another review session by Nurture Oncology. I would like to apologize for delaying this session for a couple of weeks. We were finding a new way to involve you and to make this more interesting and simplify the research articles for you to in order so that you can understand it in a better way. Today we are gonna say the application of aqueous human liquid biopsy in the field of retinoblastoma in terms of diagnosis, prognosis, and long-term outcomes. This is a breakthrough. Retinoblastoma, as you know, first of all, I'm not gonna tell you the exact title of the article. I will attach the link down, the link to the study. But the title might scare you off if I narrate it right now. But it's a really nice article to be discussed upon. To begin with, let's talk about retinoblastoma. Retinoblastoma is a childhood malignancy. It is a rare tumor in pediatric oncology, but it is the most common ocular malignancy. And why liquid biopsy is a breakthrough in retinoblastoma is because in order to do tumor biopsy of retinoblastoma before the emergence of liquid biopsy, we had to either enucleate the eye. Enucleation means remove the eye in order to just excise the tumor, or it was just too risky to put the needle, extract the tumor from the eye without seeing whether it would seed somewhere else in the eye. Suppose it's in some corner A or upside. If we do the biopsy, what if the seed gets implanted in the front? So it was very risky to excise the tumor via just a tap. Therefore, this new thing, that is evaluating or assessing the aqueous humor without even touching the actual tumor area in the eye, is a revolution in the field of retinoblastoma. Now, this article, this is the first study which we are going to study in this field, which has taken aqueous humor at the point of diagnosis, at different point of treatment, and then seeing the overall prognosis of the patients. So they're seeing how accurate aqueous humor is in terms of diagnosis, prognosis, and long-term outcome of these patients. Now, you might be thinking what exactly we are seeing in the aqueous humor. You might be thinking that aqueous humor is a separate entity, or tumor is inside the eye. So how is these two things related? So what happens is, whenever a tumor is growing, certain cells are dying, and those dying cells are being released in the aqueous humor. If I have a mesothelial tumor, suppose, sorry, I don't wish that I have it, but if someone has mesothelial tumor, then the tumor will grow, and in order to grow, some cells will die. Those cells will be slacked off in blood or in the pleural fluid. We can biopsy that fluid and analyze those cells which are being slacked off in the fluid surrounding the tumor, and evaluate its DNA, see what mutation actually caused this malignancy. This is what we are going to see in aqueous humor. We are gonna take the aqueous humor by liquid biopsy. By anterior chamber tap is the technique which they use. So see the tumor cells which were dead, and see the DNA which is out of those dead cells. So that is called as the cell-free DNA, cfDNA. I'm going to refer this term again in the video further. So here we have, I think we have 7 patients in the study. No, we have 6 patients in the study and 7 samples. How 7 samples from 6 patients? So one patient had a biocular involvement. Both the eyes were suffering from retinoblastoma. This is a study from Children's Hospital, Los Angeles, and the consent was taken. There is a minimum follow-up of 12 months in these patients, and the patients who had anterior chamber involved, those were excluded, and the patients who didn't have any anterior chamber involvement were included, because the technique is to extract aqueous humerus anterior chamber tap. Then there were 3 males and 3 females, and there was one case in which they saw germline mutation. Now, if you remember the mutations which cause retinoblastoma, one of the key mutation or the gene involved is RB1 gene. What they saw, what we are doing here is taking aqueous humor at the time of diagnosis and analyzing it via sequencing, genome sequencing, seeing that which gene is affected. Then we take it at the subsequent after the subsequent cycles of chemotherapy and see how it is, whether the mutation is still present or not, whether there is a change in the DNA, whether the tumor volume is still there. So you can go to the table 1, see the demographic and diagnostic criteria of the patients. It's a good table, you can see it. Then coming to the diagnostics of this technique, if you can see the figure 1 from the study, so we can see there are 2 cases, which is the case 33 and then we have a case 48. Now this is the diagnostic sample which we are talking about in this figure. The light blue bar is something I want to focus you on. You need not get scared by the terms single nucleotide variants or SCNA or SNV. I'm not going to explain you all what it is because that's not necessary. I just want you to know what is happening by this in the study and what is happening in the field of precision oncology and retinoblastoma. So case 1 is case 33. If you see in this bar figure, there is no light blue wala bar in it. And similarly in case 48, there is no light blue bar. They have written ND, that means not detectable. Similarly in case 33, there you can see ND, not detectable. They were not able to detect any RB1 mutation in these 2 patients. Yet these patients had retinoblastoma. So there was some other mutation that was leading to retinoblastoma, which is MYCN, MYCN, and 6p gain. Now, this is what they found in the diagnostic sample of aqueous humor. Let's go further, and now you can see the figure 2, in which we have the genomic profiles of all the patients. Again, in case 44, you can see they have marked the RB1 gene deletion, and in case 33 and case 48 look a bit different than the rest of the cases. But if we come out at the prognosis, when they did the profiling, they found that the 6p and MYCN patients suffering with 6p and MYCN gains or amplification had a relatively poor prognosis as compared to the patients having RB1 mutation. This is the importance of genomic profiling. They were able to detect this at a very early stage. Now, now another question in their mind was, in order to confirm it properly, they did some B scans to see the tumor volume. They estimated the tumor fraction, and by cfDNA analysis and a few more variables which you can see in the figure 4. In the figure, it might be difficult to just know what is this figure. Might be looking a complex one, but what important thing is, let's break it down in small parts. First is the term tumor fraction, and the second term important is tumor volume. Now tumor volume, basic thing, if I ask you what is the volume of a bottle, you will just tell me it's 200 ml or 500 ml. Similarly, tumor volume, they have this B scans via cfDNA. They measure the concentration of the cfDNA or the B scans, the measurements of the B scan, and evaluate the tumor volume of how big the tumor is in the eye. And the tumor fraction is basically how many cells are being released, or how many cells are continuously being released in the aqueous humor. So if in this figure, the tumor volume is indicated by the orange bars and the green line, which you can see. So you can see this is the figure of case 33, okay, figure 4. Tumor volume is continuously decreasing. So if a person will see via B scans, the tumor volume will be decreasing. But in reality, when we see the tumor fraction, it's constant. What does this mean? So the meaning of this thing is that the tumor, although it is the volume of the tumor is decreasing due to chemotherapy or due to any other mechanism or whatever therapy we are giving to the child, but the tumor is not getting eradicated. In the sense, it is the cells are being slacked off, then they're again growing. The cells are being slacked off and then they're again growing. So tumor fraction is constant, but the volume is decreasing. The necrosis is happening, but the increase in the cells and regeneration of cells is also having. So this is an aggressive tumor. It is continuously growing, and the chemotherapy, despite giving chemotherapy to the patient. Then see the genomic profiles. DX means diagnosis. At first, they had certain MECAN amplifications. And IVM means after first intravitreal therapy, first cycle of chemotherapy. IVM2 is second cycle of chemotherapy. IVM3 is third cycle of chemotherapy. And then ES is enucleation. So we are not able to see any changes in the genomic profile of this patient over these different stages when we took this aqueous humor sample. Whereas if you go to figure 5, now this is a responsive, this is of case 47. It was RB1 mutation. It was not MECAN or 6p gain. In this, you can see that both tumor fraction and the tumor volume is decreasing. Then if you see the genomic profiles, first at diagnosis, you can see there's variations. Now RB1 mutations are there, but after chemotherapy 1 in the genomic profile labeled as IVM, there are significantly less modifications or amplification of the profile. Whereas in IVM 2 and 3, there are even lesser than before. So this is the significance of genomic profiling and the magic of aqueous human liquid biopsy. We were able to, without even extracting the entire tumor, we were able to see the DNA response of the tumor to the therapy. We were able to see the fraction of the tumor which is still going on, and we were able to analyze that whether it's aggressive or not. We were able to see what mutation it has, and if we know what mutation it has at a very early stage, these people wanted to establish evidence. Now if we, as they have established evidence that at the diagnostic sample is good enough to show the mutation, we know that 6p gain and MECAN gain are aggressive tumors, and therefore they would need some other therapy or some other intervention as compared to the normal conventional therapy. So that's it for today, guys. I'm not going into much details of the discussion part of the paper. I just wanted to give you an overview of aqueous humor liquid biopsy and retinoblastoma, the importance of it, and what these researchers did in this paper. I wanted to break it down in small points so that you get to know how to read paper. See, I didn't go much deep into the methodology. I didn't go into the details of genomic names like SCNS and V. What I explained to you was the key results, the importance of this technique, and the explanation of the figures. So if you're interested in reading papers, this is how you can go. Just get the background of the study, see the results, and an overview of methodology of what they are doing. Thank you so much. I hope you enjoyed it. Bye bye.